So this video is for Biology 234 for the hands-on lab titled Food Safety. And the learning objectives are to culture microbes collected from fresh food sources and to relate experimental results to methods for improving food safety in your home. So you'll want to gather all your materials and then we can get started. But one thing to keep in mind, you can't do this lab until you have aged your milk 7 days. So you actually have to start this the week before. So we'll gather our materials. So we need a bottle of bleach. We need a bottle of distilled water. You'll need a digital camera or your smartphone. You'll need hand soap, which I have at my sink. You'll need isopropyl alcohol. You'll need a large cooking pot if you need to pour your plates or pour some new plates. You'll need one fresh leaf of lettuce, unwashed. And I don't think it matters what type of lettuce you get. I just got green leaf lettuce. You'll need an oven mitt if you need to pour your plates. You'll need a permanent marker. You'll need a roll of paper towels. You'll need 2 small containers of milk. And I actually had a hard time. I thought I would be able to find like pints of milk, but the smallest I could find were half gallons. But I didn't really, go to a bunch of stores to find something smaller. So you'll need 2 things of milk, and then I marked the one that I had opened last week. You'll need a source of tap water, and you'll need a stove or hot plate. And then of the things that are in your hands-on lab box, you'll need your apron. You'll need your face mask. You'll need 1 tube of the nutrient agar and 2 petri dishes, or you'll just need 2 poured plates. You'll need 2 sterile swabs. You'll need your test tube rack. And you'll need your test tube clamp. Yeah. Alright, so we've gathered all our materials. Again, you'll need to have opened one of your milks a week before. And it also suggests buying them on the same date and making sure that they both have the same expiration date. So I will just move all of this to the side and then we'll get started. Alright, so now we're ready to get started with exercise 1, which is called Food Safety. In this lab, you will incubate microbes collected from unwashed and washed produce and from opened and unopened milk. After incubation, you will compare the number and types of microbes that develop from each food source and relate the results to practices for maintaining food safety in your home. So now we're ready to get started with the procedure. It says, note, 2 containers of milk are needed. Open 1 container exactly 7 days prior to beginning the experiment and allow the second to remain closed until used in the procedures. Place both containers in a refrigerator when not in use. It is best if the 2 containers are purchased on the same date and have the same expiration date. So, just to show you guys, these both have the same expiration date, and then I did this, opened this 1 week ago, but I haven't used any of it, but this one has remained closed. So I'm gonna put these back in the refrigerator until we're ready to use them. All right, so now we can start part 1, which is inoculating plates. So step 1 says, approximately 1 hour before you begin this exercise, pour 2 agar plates. You can download the Pouring Agar Plates PDF for more detailed instructions, and I also have a video of how I did it. It says, note, plates may be poured in advance, stored in an airtight bag, and refrigerated for future use. Step 2 is clear your work area and gather all materials listed for this experiment. Step 3 is wash your hands thoroughly with soap and warm water. So I'll go wash my hands. All right, I've washed my hands, so now we're at step 4. Put on the safety gloves, face mask, apron, and goggles. All right, I've put on my safety gloves, face mask, apron, and goggles, and I realized I somehow missed that in the list of things you need to grab from your box. So you'll need to grab your safety glasses and your gloves as well. So now we're at step 5. Disinfect the work surface by wiping it with a 10% bleach solution. So I'm just gonna use my 10% bleach solution that I made and saved. And you don't need that much. All right, so now we're at step 6. Using the permanent marker, divide the bottom of each agar plate into 2 sections. Label the sections of one plate fresh milk and milk opened, and then label the sections of the other plate lettuce washed and lettuce unwashed. So this is figure 4, how your plate should look for the lettuce. And then you would do the same thing for milk. So I'll grab a white piece of paper so you can see what I'm doing. So for each of your plates, you're just gonna draw a line down the center. And then I'm going to write milk on one, and then I will do lettuce on the other. And then we'll want one side to be washed and one side to be unwashed. So I'll just do it the same on both plates. Or milk will be opened. And then, so lettuce would be unwashed. And then milk will be fresh. So for my lettuce plate, I have one side for washed and the other side for unwashed. And then for my milk plate, I have one side that says fresh and then the other side that says opened. So now we're at step 7, and it says remove a sterile swab from the packet and moisten it with distilled water. So you'll want to grab your swabs, and then I don't think it mentioned this, but I'm going to grab a plastic cup to put my distilled water in. And you don't need much. You just need enough water to soak your swab in. So for step 7, I will just put one of my swabs in the distilled water and keep the other one closed in its package. And then the next step is step 8, rub the moistened swab on the surface of the unwashed lettuce. So I'll go grab my lettuce.